All right. Good morning, everybody. Welcome to the Ancestry and Diversity Working Group Forum. We're excited to have you all here today. Dr. Alex Ioannidis is going to be speaking to us today, and Alex is stepping on as one of the chairs for the ADWG, so we're all excited to hear about his work. Dr. Ioannidis is an assistant professor of biomolecular engineering at the University of California, Santa Cruz, and a member of the Genomics Institute. He also teaches classes in artificial intelligence and data science at Stanford. He earned his PhD in Stanford in computational and mathematical engineering, together with an MS in management science and engineering. Graduated summa cum laude from Harvard University in chemistry and physics, and earned an MPhil at the University of Cambridge from the Department of Applied Math and Theoretical Physics and Computational Biology. His research focuses on the design of algorithms and the application of computational methods for problems in genomics, clinical data science, and precision health, with a particular focus in diverse worldwide populations. Alex, take it away. We're so excited to have you here today.
Hi, Clarissa. Thank you so much for the intro. It's great to be here. So I'm going to share really quickly. Hopefully this works. Everyone's able to see these slides. Looks good. Great. Okay, so I'm going to be talking today about genetic ancestry. So I'm going to talk about some algorithms and applications of genetic ancestry. I want to point out before I start, though, of course, that this is only a piece of the puzzle behind ancestry and diversity. There's so much more. In fact, more than 50% would be all sorts of things revolving around identities and social and environmental factors. And so I don't want to lessen that by the fact that this talk is going to happen to focus on genetic ancestry. But I think genetic ancestry is also interesting, and it's interesting when we're talking about, of course, genetics as we care about in ClinGen, and the interplay between genetics and other factors. So if you take a look at this title slide, what you'll see is one representation that we frequently use for genetic ancestry, which is called an admixture plot. Comes from the program's structure originally, which was actually created by Jonathan Pritchard, who's at Stanford. There's been a lot of modifications since then to scale these algorithms and to increase their accuracy, speed, et cetera. So what we're looking at here is basically every vertical sliver in this plot, and you can see they're really tiny slivers because we have so many people in here. Every vertical sliver is a person, and the color along that vertical bar for that person represents the percentage fraction that they have been assigned genetically to the different colored ancestry clusters. So I'm showing here just a cross-section of the UK Biobank, and you might wonder why there are so many British samples in the UK Biobank, and because people have moved to the UK and were included in the study. So these countries are the places of birth that were registered for the people in the UK Biobank. And so you see a lot of colors. So there's some diversity even in the UK Biobank, although you'll see in a moment that is, of course, mostly British genetic ancestry. But we can still see, some diversity popping out, and we can start to see some patterns. So this is an unsupervised clustering algorithm. So there's no label attached to any of these colors, any of these genetic clusters. We impose our own views of what we would like to call them. For example, you'll see that there's a green cluster that's present basically in countries in the Americas. So you see it here in Mexico, and you see it in Chile, and you see it in South America, and these are generic. South America is just self-reported birthplace South America. So and then self-reported birthplaces in Peru. So we have a supposition that this might be an indigenous American ancestry, and that is actually what it looks like. And so these are Latin American individuals who have some indigenous American ancestry, but there's no specific label that we can apply. This is, again, unsupervised clustering. You'll see a couple other interesting patterns here if you look a little more depth. The other thing you'll see in the Americas is this red cluster, and that's going to be the Spanish European-like ancestry. But you'll see it all across these other countries as well. You see it in Zimbabwe here. And you might wonder, why is there so much European ancestry in Zimbabwe? And again, remember, these are individuals in the UK. They're in the UK Biobank. So the people who were born in Zimbabwe, but they moved to England, and so there is a selection bias in who's selected. And so, of course, we have maybe a lot of British people who may have been born in Zimbabwe to British parents, and that's why they moved back to the UK. So that's why you see this large European ancestry there. But other interesting social things can pop out as well. You'll see in Uganda and in Kenya, the single largest ancestry type that we have is this orange ancestry. And there is some of the red European, as I already mentioned, but there's a lot of this orange, and then also in Tanzania. And the other place that you see orange, you'll see it in Afghanistan. You see some of it in Myanmar. It's actually, I'll just tell you, if you were to look at the whole spread of the different countries, it seems to be a South Asian ancestry. So think of like India and Pakistan, and you might wonder, why is there ancestry from India and Pakistan in Kenya and Uganda and Tanzania? And the answer is actually, again, due to British colonialism. A lot of people from the subcontinent, from India, moved during the British colonial period to British Africa. Gandhi is an example. He was a lawyer in South Africa. And after decolonialism, after the British left, many of these people had to leave as well, and many of them went to the UK. And so that's, again, selection bias. We're looking at the UK Biobank. So we see people born in these countries, but who happen to be in the UK now. So we can start to see some interesting social things too, just coming out of the genetic clusters. And that's sometimes interesting if that's, the data that we have. This now looking at the entire UK Biobank, again, as I said, most of the individuals are going to be this blue ancestry, which is a sort of Northern European, British-like ancestry. So it's only a small set that actually are born outside the UK. But anyway, we are getting more and more diverse biobanks now. We have all of us and others coming online. And so these sort of diverse ancestry algorithms start to become more important. And this is an algorithm that we had developed that scales really well to these large biobanks. And you can run it in only a day rather than the existing algorithms take like a month. So I don't want to go into that too much more depth, of just intro to what we're going to be looking at with genetic ancestry. And then I'm going to show something else here that's a bit out of left field, but just maybe to color the other side of the equation and how we need to be careful about thinking about all of these topics, because there's really useful things we can get, and we can start to tease out these social and historical phenomenon and look at differences in allele frequencies, which I'll be talking about. But there's also all sorts of ethical issues we have to think about, and I just want to put this up here as like an exclamation point or a red flag, right? And this is an interview which I'm sure many of you haven't seen, maybe you have. But it was between Tucker Carlson, it was about 2 months ago, and Vladimir Putin. And the only reason I'm putting this up here is to point out that about halfway through the interview, very oddly, Tucker asked Putin what were the two sort of scientific advances that he thinks will be most influential in war and strategy and other geopolitical things going forward. And not surprisingly, Putin said artificial intelligence. I think we'd all have expected that. But the other thing he said was genetics. And I think that's just a warning to us all that this is a field. It's not just about research. It's not just about improving people's lives. There's all sorts of other red flags that can crop up here, and we need to be careful of that, right? So I don't want to say more about that. But just to throw out another one of these red flags that's more directly related to what I'm going to be talking about algorithmically. And this is a figure number 2 from the All of Us paper, which came out also around the same time, about 2 months ago, and was roundly criticized, and rightly so, for having quite prominently conflated race, which you see labeled right here in the top of panel A of this figure, and genetic ancestry, because what is plotted in this figure is genetic ancestry. It's just the sequence data that's been analyzed. But the two are being conflated here, and as we know, that is very problematic, and a number of other problematic things happen here. An algorithm was used called UMAP, which I'll discuss briefly, which is designed to accentuate differences in clusters. And there are applications where one might want to do that, but it's not clear that one wants to accentuate and distort. It's a nonlinear algorithm that actually distorts the data. Distorted the data to try to make it look like the different groups, which you're now labeling race, are more genetically different than they are. So this was very problematic, and there's a lot of other things we can talk about here in terms of the labeling and the categories that were used, et cetera, that have issues as well. But you see anyway a plot here on the right which is a rotated version of the plots we were looking at earlier. And so you see these are quite common, these admixture type plots where every individual here is going to be a row. You can see how many individuals there are, just lots, again, because this is all of us data set, quite large. And you see the colors are these clusters. So we'll talk about this a little bit. The algorithm that was used here is one that has some issues as well, and I'll mention that in a minute. And I just threw this in here. So this is the discussion. Jonathan Pritchard, who's at Stanford, had pointed out the dangers of using this UMAP and then using it to accentuate differences in this context, really not great. And then Graham Coop had pointed out some other issues, which is the conflation of genetic ancestry with race and ethnicity. And as I mentioned, another problem in this figure. And then all of this, piggybacks onto existing controversies that exist in our algorithm community between people who are really against certain algorithms. In this case, a professor here at Caltech who's long been against UMAP in any context, although it's widely used and quite productively used in single cell and other modalities. And a professor here at McGill, Simone Gravel, who has advocated for UMAP in certain contexts in genetic ancestry. And as I said, it may be fine to use depending on your purpose, but certainly labeling race is a problem. So I'm going to talk now just to give an overview. So what are we talking about? UMAP and these admixture plots. What is all of this? So all of this comes down to basically dimensionality reduction. So when we're looking at genomic data, we've got many positions of variation, right? Millions of SNPs, can be more than that if we're looking at whole genome sequences. And so we can represent these millions of positions, the variation, in a million-dimensional space. And then we have a problem if we're trying to visualize. So we need to do something to represent this diversity in only 2 dimensions and get our figure in our paper. And so one thing that we can do is what's called dimensionality reduction. This is really nice set of techniques, of course, where you can project points here. I'm showing from not a million dimensions to 2, but rather just from 3 dimensions to 2. But so we have 3-dimensional data. Here the points are colored differently depending on the cluster they belong to. And we just project these 3-dimensional data points down to the 2-dimensional crosshatched plane you see here. So some plane is chosen, and then we cast the shadows down of all the points onto that plane, or up if they're below it, and we get a 2-dimensional representation, which seems to pretty much preserve the structure of the data. You see that most of the clusters are preserved, et cetera. We've lost a little bit of information in the casting of the shadows down onto this plane, but in general, we've retained most of our information. So this is dimensionality reduction. And in this case, it's principal component analysis, because the plane on which to project was chosen to be the plane that captured the most variation in the data, which would seem intuitively to be what we want to do. We want to find a space that captures the most variation. Just throw this in, because this is where all these debates really stem from, the fact that when we collapse dimensions, even just from 3 to 2, we distort the data. We lose some information. And this book, which I quite enjoy, actually, it's a fun read. The author carved out this 3-dimensional wooden shape, but if you casted shadow down into only 2 dimensions, you get different letters depending on which 2 dimensions you choose. So you get a B, a G, or an E, and the question is, what is this shape actually? Is it a G, an E, or a B? And of course, it's all of them because it's a 3-dimensional shape, so it can be all of them. And so dimensionality reduction, although it allows us to visualize the data in very high-dimensional space, it loses information, and that's inherent. And so there's some distortion that takes place inherently. And that's where all the problems begin. But it's still a useful technique. As I said, if we're going from million dimensions down to 2, at least now we can sort of see things in the data. We can't visualize a million dimensions, although we're obviously going to lose a lot. And as I mentioned, the most prominent version of dimensionality reduction is principal component analysis, which has been widely used from early days of array data in genetics. And we have this well-known paper, which is Genes Mirroring Geography, where we do a principal component analysis of individuals. Every one of these letter codes is a different person, colored by the country they came from in Europe. And really just projecting their genetic data, in this case was hundreds of thousands of SNPs, down to just 2 dimensions, still preserves basically the almost geographic structure of Europe. Even though no geography was in here, this is just genome sequence data. So kind of impressive at one level, and it shows that there is some ability to capture diversity with this technique. And you see, even this is I always found interesting. If you zoom in on Switzerland, which is right here, CH, you see the structure even of the different language communities in Switzerland being somewhat elucidated. You see that you've got the French-speaking Swiss are these green squares, you've got the German-speaking Swiss are the red diamonds, and you've got the Italian-speaking Swiss are the blue triangles. So you see actual separation even within a country, depending on the region and the language that people are speaking, which is kind of impressive. And this I just throw it out here because from the Stanford side, this is a paper that even made it into one of the stones that are outside the Stanford Medical School here, commemorating the studies that have been done at Stanford. So sort of an iconic figure in any case, representing basically genetic ancestry and what we can do with algorithms. But again, principal component analysis is just one method of dimensionality reduction, and we can see it can be problematic. And here is a really simple example of that. You've got two clusters here. The first principal component is going to be the direction of greatest variation in your data. So that's going to be this horizontal axis right here that separates the data as much as possible, the greatest variation. And if you were to project all your data just onto that one axis, this horizontal line, you would still have two clusters. This data would project on this side, and this data would project on this side. However, if you do the same type of analysis, principal component analysis on this data, you're going to get a line that's a diagonal line, because that's the direction of greatest variation of your data, running right up along here. And if you project all of these points onto that diagonal line, you're going to get just a long smear. These points are going to project right on top of each other onto this diagonal line. And you will no longer see that you had two distinct separated clusters. So you will have lost the structure in your data set. You won't be able to visualize it. You won't even know it was there if you never looked at the higher dimensional space to begin with, which is going to be the case for genetic data, because you're never going to look at the million dimensional space that it starts out in. So you just wouldn't even know that the structure was there. So PCA can lose things, as we mentioned. This is the problem with dimensionality reduction. So, okay, so can be problems. So we look for other methods. So this is a method that we developed recently called, archetypal analysis of population genetics. And we use the algorithm that's behind principal component analysis, which is called the singular value decomposition, but we don't actually do a projection. So we don't lose any information. And this is important. So we use the algorithm to speed things up, but we don't actually lose any information. And then we do a clustering method called archetypal analysis. And this two-step process allows us to handle really, really large data sets because we do this matrix processing. But it prevents us from really distorting the structure of the data. And so this is an algorithm that is very related to, basically is very similar to the one used in this paper. It's basically the same in terms of how they get their computational efficiency. However, this is something we did about 2 years ago. The issue is that it still is not that efficient at very large data sets, and it still has some other things that could be improved. So we have developed a neural network-based algorithm which produced the plots I showed in the title slide at the beginning, which actually allows us to skip the whole pre-processing and just process the whole data set in one go. And it also allows us to do something kind of nice, which is that when you do these sort of plots, and you can see it here with the different colors, and here again with the different colors, you have, although this is unsupervised, so again, the clusters emerge organically. You do not stipulate what ancestries you want the algorithm to find, which is sort of nice. There's no sort of, there's nothing being inserted in from our conception of identities in the world. You still have to choose how many colors you want, basically how many clusters you want. And so usually the way this is done is that you try to rerun the algorithm multiple times with many different numbers of clusters. You try to subdivide the data into 2 clusters and 3 and then 4 and then 5 and then 6, et cetera, and see what it looks like. And what's nice about the neural network is because it has this embedding space, we can actually do all of the different numbers of clusters in one go. There's a lot of shared information, as you might imagine, when you cluster different numbers of clusters. And so we can harness that shared information and grab it all together and plot in one go. And so this really speeds things up as well. So instead of rerunning the algorithm multiple times, you can do it really quickly. So again, now I'm going to sort of take a step back. This is sort of the motivation of why we're doing this, to try to be able to start, understanding the structure of our data, try to allude to some of the pitfalls that can come up, and some of the computational issues. And now I'm just going to step back for a minute and remind people why we're doing this and what's the basis. So we have, of course, human populations extremely similar to each other genetically compared to other species. This is really important to keep in mind in all of this. We're trying to, identify some, small amounts of variation, but in general, we're extremely similar. We all went through bottlenecks inside Africa, of course. So a lot of shared ancestry after those bottlenecks, and then a bottleneck for those populations that came out of Africa. Spread around the world in the last few tens of thousands of years, eventually another bottleneck coming into the Americas and then settling the Americas as well. So each of these groups, once located in a different area, begins to drift genetically on their own. Allele frequencies change with time. They're executing sort of a random walk. Of course, there's continuous admixture and interaction between the populations, which adds more complexity and reduces the overall divergence of populations, because they're not completely isolated. But with time, there is some change in allele frequencies just with random drifts. Of course, most of the variants were present from the outset, in all humans from their time leaving Africa. It's just a question of the frequencies of those variants and how they've changed with time in different populations. And we have equations to describe it. I'm not going to go into this, but it's the point is that we're basically executing a random walk in which every population has here 3 different variants. So the frequencies of all of them are changing independently. And when populations split, they begin to execute their own random walk in this frequency space and begin to slowly diverge in frequency space and accumulate different frequencies in each of their alleles. Here, just 3 alleles being shown. And so we can represent that with sort of a tree that just represents the change in the frequencies as the populations accumulate. And we can do statistics on this, and I won't get into it in too much depth. But the result of these change in allele frequencies is what we see in these principal component analyses, and here I'm showing it in a 3-dimensional space, is that each of these little dots is a different individual, and they have accumulated, depending on the population they're in. The different worldwide populations here from different continents are colored different colors. They have diverged in this frequency space as they accumulate differences in frequencies. And so we can find the clusters in this frequency space, and that's these bold dots. And once we find these cluster centers, and you can see this is just sort of somewhat arbitrary where exactly a cluster center is going to lie, but this is something the algorithm will choose to try to fit the density of the clusters as well as possible. We can assign every individual, every little dot, some fractional identity to each of these bold cluster centers, depending on which ones it's closest to. And if it's in between 2, it's 50-50. And if it's close to 1, maybe 70-30, et cetera. And it's that assignment, determining the cluster centers and then assigning fractions, that gives us these plots. And so I'm not going to go into the algorithm too much in depth, but suffice it to say that all of these methods use somewhat the same basic mathematical model, but then the question is how to optimize it. And different optimizations get you different performance. And the one that I've been discussing that we've looked at plots for gets you this wonderful scaling, whereas the number of samples increases. The execution time really doesn't increase much at all. So you can run for huge numbers of samples in a biobank without it taking very long. And most of the other existing algorithms, they start taking a long time to run. So that was our contribution in that paper with the neural admixture algorithm and the preceding one, the archetypal analysis. So it's a computational speed up. And as I mentioned, we can do all different numbers of clusters in one go. And that's depicted here. Inside the neural net, we basically have an embedding space so we can cluster many different clusters all at once. So that's another speed up. And we can get fun things out of these clusters. I'll just throw this up here. Once we've done this clustering, here I'm showing individuals from different islands all across the Pacific. Here see the different islands. And one thing you see when you look at these ancestry clusters, we've seen this plot now before, is here on Easter Island, Rapa Nui, we see this green cluster showing up. And the only other place we're really seeing green is in the Americas. And it's, again, it's an indigenous American genetic ancestry. And there's a question, why is that indigenous American genetic ancestry on Easter Island? And it, you know, turned out to be a really interesting story, actually, why it was there. So this is something you can do, and you can visualize these clusters in other ways. So I'm going to show you here. This is a compositional analysis. So basically, every point is a person again. And where the person lies inside this triangle tells you basically their fractional ancestry on each of these clusters. So if they're, you know, mostly Polynesian, they're going to be over near this vertex, mostly European here, and mostly Native American up here. And the fact that the individuals in this case, and their people in this case from this island, this Easter Island, the fact that they're all lying along this dotted line tells you something about the order in which the different genetic ancestries combined on that island. In fact, what it tells you is that the Polynesian and the indigenous American ancestries combined first, and that the European came in later and essentially diluted out the other two at the same rate. And that's why you have this line. So it tells you that the European came later, which is really interesting if you think about it for a little bit. And so you can do some fun math on that. And you can also do these plots in higher dimensional space. This is a tetrahedron where we have got 4 different ancestries, and the points are inside the tetrahedron. And you learn a little bit more about the population history in that case. So the other thing we can do, everything I've talked about so far, is genetic ancestry as a percentage. So giving somebody, an assignment to a cluster where they're 20% this cluster and 80% that cluster or something. We can also assign genetic ancestry based on segments of the chromosome itself. So we can, assign every segment of the genome to an ancestry. And this is going to be very useful if you have admixed populations who have, a mosaic of different genetic ancestries. And here I'm showing, again, an individual who is, you can see one parent was this lower chromosome completely European, the other parent has a bit of a mix. And we can do many things once we've identified these different segments. One of them is to look at the allele frequencies in the different ancestries. So that gets quite interesting for applications related to ClinGen, and I'm going to be talking about that in a minute. We can also look at the length of these different ancestry segments, and that tells us things about when these different populations mix, because recombination breaks up segments with time, of course. And so if they mixed a long time ago, the segments will get smaller, and you see that here. Kind of small segments of indigenous American ancestry is indicative that this ancestry came into the Polynesian ancestry a long time ago, which is interesting again. So this is an algorithm that we developed. I'm going to talk about it much, but to do this local ancestry, there's several algorithms out there. This one is quite fast, but there's a number out there. And the way they really work is they look at the code along the genome. You can binarize it by basically categorizing every position as being the reference or the alternative allele. So you can have 0 or 1, and then you can apply your favorite machine learning methods on top of it. In this case, we implemented it with tree-based methods, but SVMs also were implemented in our package, and also a host of other existing machine learning methods, and then you apply some sort of smoother on top, conditional random field, or HMM, or something of that sort, and then you get your final assignments. And so this is a nice algorithm developed by 2 master's students actually at Stanford who worked with me on it, and I highly recommend for people looking for a local ancestry, you might try it out. The other thing that we can get out of it is that we can improve the phasing. So if you see that if you have an ancestry switch that occurs from, let's say here, the blue to the red ancestry, the exact same point that the other chromosome switches in the exact opposite direction, the chance of this happening, coincidentally is basically 0. If you have the exact opposite direction switch at the exact same point, this is actually just because there's a phase error. These 2 segments should be swapped. And this is just from when we did the sequencing, right? Shotgun sequencing or array-based, we don't actually know which of the 2 chromosomes any variant is coming from relative to its neighbors. And so improving phasing is something we get for free out of this algorithm, and it's something that's important if we're looking at long haplotype-based markers or repeats or something like that. And so that can be useful. And here we're showing that phasing playing out. So the algorithm goes and fixes the phasing. You can see these different colored segments that were swapped, and now they're properly aligned. Okay, so what else can we do? So now, once we've identified every segment of the genome, and here we see one individual, but what we're looking at are here 22 autosomes, and what we're doing is coloring every segment of their genome, again, in terms of which of the ancestries it came from. And so this individual from Latin America has European, Native American, and Polynesian segments. So this is actually another individual from this island that's ruled by Chile right now, which is called Easter Island. And this ancestry sort of mapping will play out throughout a lot of California and the United States in general, and in Latin America, where you'll have often European, Indigenous American, African ancestries, but it could be East Asian ancestries also. In Hawaii, where we're doing analyses now, we have really an incredible mix of East Asian, African, Native, Indigenous American, actually quite a bit of Hispanic ancestry through Pearl Harbor and the bases there, in European. So you get a real mix in each individual, and then you can look just at the segments from one ancestry, and you can look at where they project in these PCA projections that we were discussing earlier, and visualize which regions even from within Switzerland, say, or within Europe, they're in European ancestry comes from, and do the same thing with Native American and others, et cetera. And it involves some sparse matrix methods, which allow you to deal with what is effectively missing data when you're analyzing any one of these ancestries alone, because you're missing the other segments of this individual's genome that were from another ancestry. So you've got to account for that missing data. And so I'll just show you what this looks like, just so you get an intuition for it. So here we have individuals again from Pacific Islands, and we're overlaying them. They're in text here. We're overlaying just their European ancestry on a PCA of individuals, and these are these colored dots from different countries in Europe. Okay, so we've got Ireland and the United Kingdom up here. We've got Spain here and Italy here. So we have these different countries in Europe. And then if we overlay the European ancestry of the islands on top, we see most of the European ancestry from the French Polynesian islands is lying on France, right? Which is not surprising, because that's French colonial ancestry that mixed into the French Polynesia. However, we see a different pattern, so that's why it's marked differently in these green diamonds. The green diamonds are individuals from Easter Island, which I mentioned is part of Chile, and so you would expect that the European ancestry there is quite possibly going to look more Spanish, and indeed it does. These individuals are overlaid on top of the points from Spain. Keep in mind that all of this is, for these islanders, specific just to their European ancestry segment. So we first had to look at doing this sort of local ancestry along the genome for them. If we were looking at their entire genome, they would not project anywhere within Europe, because much of their genome is Polynesian, and so they would project somewhere far, far away. It's the fact that we've deconvolved these two different ancestries, and now we're looking just at the European, that allows us to then zoom in and see where that specifically comes from. So we can do these sort of analyses, and now we can do this same thing, but just with their Polynesian ancestry. And if we look just at their Polynesian ancestry, we can get what you'll see here in the PCA, separation of every island from every other island, because there's founder effects, and each island has its own characteristic signal. And the founder effects are going to be really important for allele frequencies, because, of course, when you have a founder population, a small number of individuals coming to a new island and then growing to a large population, the allele frequencies on that new island are going to be characterized by the alleles that the founders brought, which is going to be a real, huge genetic drift randomization process in terms of changing those allele frequencies from what they were in the home island. And you can see that here in a plot, which is ancestry specific, so using this sort of methodology, but now overlaid with something basically like UMAP, which again, as I mentioned, accentuates differences. And you can see that here, the PCA, you can see it separating out some of the islands, but not in such an accentuated way. Now we can really see the accentuated separation, which is useful if we want to be able to, let's say, do some sort of QC or identify exactly what island someone came from. But if we're trying to just represent how basically similar we are, this is not the method you want to use, because it's accentuating differences. And so I think that's, again, the pitfall that people fell into on the All of Us paper. I mean, we can do this across all of Oceania and Southeast Asia and East Asia, and I'm not going to go to it in depth, but you get really pretty plots. And this is from a much larger data set I'm working on now that covers all of Southeast Asia and Oceania. Many populations, and just all incredible diversity from all of these islands and different ways of settlement that have gone on. And here we're looking two different ancestries that people don't think about that much when they talk about Oceania. But there are two very distinct ancestries in that region, and one is this Austronesian, so you can think more Polynesian ancestry, and one is Papuan, which is the ancestry you'll find on Papua New Guinea and in Aboriginal Australians, which is a very different historical ancestry that came in the earliest out of Africa expansion into the region, and really predates by a long time the more East Asian-like ancestry that spread out ultimately from Taiwan, that spread through Polynesian islands and characterizes Polynesians today. So we can do these sort of things in other parts of the world. So we can do them in Mexico. And here I'm showing population diversity of different groups in Mexico, labeled by the different indigenous identities. And you can see separation even within the Mayan-speaking regions. We've got separation from the groups in Guatemala that are Mayan-speaking versus those in Mexico. And keep in mind, again, this is looking just now at the indigenous American genetics, at just that component. So we're not talking about whether some groups have more Spanish ancestry or not. We've separated that out completely, and we're still looking at diversity basically based on, historical divisions that existed even back in ancient times between the different Mayan-speaking groups and the city-states throughout the Yucatan, et cetera. And so how does this algorithm really work at its heart when we're separating out these different ancestries? And I said we're going to be missing large sections of the genome because we are looking only at the European segments, or we're looking only at the indigenous American segments. So what we do is we utilize the fact that we can compute distances between any two individuals, even if large sections of their genome are missing, by comparing the sites which are not missing in either of them. And so we count basically the number of differences we see in their variance. So reference versus alternative across all the segments that they do share, and we get a distance, which we can then just normalize based on the total number of positions that we were able to analyze for them. So basically the number of differences per, let's say genetic unit, right? And so now we have a distance. And so we have a distance between every individual and every other individual. And so that lets us make a distance chart, right? And here I'm showing a distance chart from sort of old road maps that you used to have before Google Maps, which is where you would have in the bottom right-hand corner of the folded up road map, the distance labeled between prominent landmarks on the map and other landmarks on the map, so you could read off how long it was going to take you to do that drive. And the reason I'm showing this is the question now is, if you have this matrix of the distances between every prominent position on the map and every other position, but you don't have the map anymore, you just have this little matrix from the bottom corner, it got ripped off, let's say, can you reconstruct the entire map just from this matrix of distances? And the answer is yes. And there's an algorithm to do it, and it's called MDS. And that is what we use now for these genetic samples. We have the distance of every sample from every other in that particular ancestry. And so now we can recreate a two-dimensional map, even if there was never really a geographical map, let's say, but we create one, and that's how we get these two-dimensional plots. And that's exactly how we got this plot. And so this is a method that we've developed that also allows you to have many different types of arrays or sequencing technologies overlaid, even though they will overlap and here I'm showing the intersection of the number of sites for all of these different particular arrays. They may overlap at only a very small number of sites, but each of them pairwise overlaps at, of course, a larger number of sites. And so all you need are the pairwise overlaps to compute these distances. And so you can actually compute the distances really nicely for many, many, many different sequencing technologies, without having to look at the overlap of all of them, which might be quite low. And then once you've got the distances, you can reconstruct this map. And so that's a nice little technique. And we can, again, be missing lots of sites because there may be different ancestry. And we don't want to consider that ancestry right now. So we have lots of missing data, and it will still work. And again, lots of missing data means we'll have even fewer sites in the overall overlap, but that's okay because we're going to look only at pairwise between each of the different technologies. And we have this method that's on GitHub. You can use it if you want. That allows you to make these plots based on this algorithm. So that's another ancestry-specific thing that we've worked on algorithmically, and we've gotten some nice things out of it. Here we're looking at the different segments just from East Asian ancestry across the different states of Mexico, and you can see in Guerrero, which is Acapulco, you have actually quite a bit of East Asian ancestry. It's still quite small, but you have it. And this is because this was the main port that the Spanish had for trading with East Asia, with the Philippines, basically. And so we're seeing the effect of that trade here, and we're actually able to identify the ancestry that contributed to it, and we find it mostly coming from here in the southern Philippines. And it makes sense. These were the Manila galleons that the Spanish would sail across the Pacific, and they would come down through Monterey, actually, would be their first stop to take on water, and then they come down along the Pacific coast into Acapulco. And we can see that. And that was great work I did with Juan Esteban Rodriguez, who was a master's student at the time, but now he's a PhD student at Stanford. So great student. And so all of these algorithms I'm talking about are on our GitHub, and anyone who wants to play around with them can download them and run them, and we've tried to make them all interoperable and work really nicely with VCF files, et cetera. And so we're working now more on deep learning applications in this space. So we're working on algorithms that allow us to have very few references, because from these more diverse populations, we're often missing whole genome sequences. And so it would be beneficial if we could have an algorithm that can learn a lot from the databases where we do have a lot of whole genome sequences, and then try to apply some of that learning. So maybe these sort of patterns that you see of haplotypes and things like that, to then port over to the more diverse genomes that we don't have so many of, so that we can do some transfer learning and get as much as we can out of those. And so this is an algorithm to do that. And so with a great master's student that I worked with on that. And now this is what I really wanted to focus on. So this is a transformer-based algorithm that we have developed to get away from the whole concept of categorical ancestry. And so I think this is really important now that we're thinking about the fact that ancestries are not genetic ancestries, are not clear blocks, European, African, East Asian. That's nonsense. Of course, there's just a continuum of allele frequency changes across the landscape, and that's why PCA captures these really nice gradients and continuum very nicely. We'd like to port that sort of a view to labeling segments of the genome, because it is very useful to label different haplotypes and segments of the genome. And the way to do it, we've decided, is again with a transformer deep learning approach, where we can actually label every segment of the genome. And here you're looking just chromosome 22 of an individual based on the coordinates of where that genetic ancestry comes from. So now we have a continuum. It's just coordinates. So coordinates can be visualized as latitude and longitude. So where would that segment map to the most similar population in terms of where populations live on the globe? So you can see this segment is mapping to around Nigeria, and this blue segment is the different segments are represented by different dots here. So the different windows are mapping to Spain, and some of them to southern France. And then here, these segments at the end are mapping to some of them to Vietnam and some of them to southern China. So that's nice, although of course geography is just some sort of strange proxy for genetic ancestry. It doesn't really have anything inherently to do with it, so maybe we should get rid of geography entirely. And I'll talk to you about that in a minute. But here, just to show those dots, so here's chromosome 22 for this individual, sort of growing up along the z-axis, and every point in purple along that chromosome, every segment of the chromosome where it's being predicted to have originated in terms of the coordinates on the globe. So you can see that now most of these segments they're growing right now originated in Europe, and now we've gotten to the end of chromosome 22, and those segments, these purple segments, seems to be predicting as coming from somewhere in East Asia, and the early ones were coming from Africa. So when we can do this with other individuals, and it works actually quite well. It's really surprising. And now we have a way that we don't have to label. I'm coloring here because we're just so used to thinking that way. I'm coloring here the chromosome by the different continents that these segments originated from, but we can get rid of these colors altogether, and we can just talk about the latitude and longitude that every segment seems to have originated from. But as I mentioned, we can go a step further and get rid of latitude and longitude, which is just a weird proxy of genetics. Geography is a different thing, really, right? And we can put coordinates just from PCA space. So we can just put coordinates for every section of the chromosome in terms of where that section of the chromosome would have originated in terms of a generalized PCA space. So where would that window have come from in principal component coordinates? And we can do this for all of the 1,000 genome samples, and that's what I'm showing here. Here I'm showing every segment, every piece of every chromosome of every individual in 1,000 genomes, and where they're predicted to come from geographically, and where they're predicted to come from in terms of a PCA space. And you just start to see this beautiful continuum starting to emerge, which is just much more accurate than these sort of categories that we've used for Ancestry in the past. And I think this is really the way forward, and we're calling this this G3net, Genome Geographical Genetic Network, so G3net. And this is great work that was done by another master's student at Stanford, Richa, who was a computer science master's student, and now she's doing her PhD at Cornell at CS. So I had some slides just to talk about the one extra piece of genetic ancestry we've got, which is identity by descent segments. So these are segments that you have here, you see in orange, because you share the same ancestor in your pedigree. So here's an ancestor in your pedigree at an orange chromosome. That ancestor is shared with both this descendant and this descendant, and so sure enough, they each inherited one small piece of this chromosome in orange, identical by descent, because they descend from the same person. There are other segments of their other chromosomes are from different individuals in their family trees, but this one segment they share identically from the same ancestor. And so how do we identify these? Well, they're quite straightforward. If you share a segment identical like this by descent, it has exactly the same sequence. And to see the exact same sequence of base pairs for a long enough run is not going to happen by chance. And so imagine seeing exactly the same sequence of heads and tails, like heads, 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 tails, 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 right? In two different records. You can't get that exact, let's say, the same exact heads and tails sequence perpetuated for 20 flips in a row by chance. That's never going to happen. So it must be that this is a record of the same exact instance of flipping, and that's how we identify these. And with these segments, looking at how long they are, we can figure out relatedness, and these are something that people enjoy going onto these consumer DNA companies to do and figure out networks. But we can build whole population-wide clustering networks, and that allows us to do really fine-scale analysis of where individuals are connected in ancestrally. And this is where a lot of this fine-scale analysis comes from, and also really gets into all sorts of issues of privacy, of course, and other things. Certainly the FBI is using this, and so there's definitely privacy issues, and also issues of which populations have their DNA sequences in these law enforcement databases already, and there's certain populations that are more heavily affected by that, and that causes bias in how these matches really are ascertained. So there's all sorts of issues around that, and there's also issues in terms of security and privacy in sensitive ethnic groups. And here I'm giving an example in China from the Uyghurs. And so all of this sort of stuff comes up mostly more with identity by descent segments than it does with ancestry. Ancestry is much broader, but identity by descent really, you can triangulate an individual directly into their family, et cetera, and even the town that their ancestors came from, and this starts to become an issue. And I'm showing it here again in that example I've been showing throughout of these different islands. We can see how the islands are connected, and we can do these IBD segments now, but we can look at them in different ancestries. So we can see what's this network look like just in your European ancestry, and the network may look very different than it does in your, let's say, Pacific Island Polynesian network, right? So you see here the islands are connected. Here we're looking at IBD segments, but just in the European ancestry for these islanders, and you see that the French Polynesian islands are connected, but they're not connected, the French Polynesian islands, into Easter Island, because this European ancestry is different on those islands. So the European connectivity looks very different than the Polynesian ancestry connection between the islands, where all the islands are connected, because of course they're all Polynesian and shared the same ancestors when they were founded. So we can see very different patterns there also. So that's kind of interesting. And those different patterns we use to reconstruct these sort of settlement patterns, and that's work we've done. And we can also date exactly when they were each settled using these segments. So I won't go into that in too much depth, but using these segments, lengths, and here we're plotting on a log axis, we can figure out as a recombination how long ago the segments came from a same ancestor, et cetera, et cetera. We do some math, and we can figure out when every island was settled, basically. And we can do this sort of thing also with populations in the New World that have come due to colonialism, and figure out when the different groups mixed. And so here I'm showing the Mexico Biobank, and you can see again, a plot of the type we've seen before. Every vertical line is a different person. We've got lots of people in here, and we can see just a whole spectrum of the red Indigenous American ancestry overlaid on the European ancestry. So there's just a whole spectrum across Mexico. It's really, really a diverse country. And we can do these sort of analyses there. And again, this biobank comes from all across every state in Mexico, actually every municipality. It's something I worked with a collaborator who was the leader of all of this work in Mexico, Andres Moreno, and I worked with him for many years. And this now allows, if we apply these sort of genetic ancestry algorithms onto this sort of a data set, we get really fine resolution information. And I'm just showing one figure here from our paper on the Mexico Biobank, showing all the different kind of structure and information you can get out of it. I don't have time to go into it in depth, but we've seen some of these figures already. You've seen this sort of figure. This is a Admersher plot, and these are our UMAP plots. But what do we care about? Well, let's look at the ancestry-specific allele frequencies. So now we can look at allele frequencies, and these are all for farm GKB variants across Mexico, right? And we can see that there are really large frequency differences even within Mexico, right? So we would naively think that maybe what we need to do are categorical descriptors. Do you have Latin American ancestry? Are you Hispanic, or are you white or African American, et cetera? And here we would see that even if we had a descriptor that was called Mexican specifically, we would still be missing a lot of allele frequency differences because there's large differences between the North and the South. And now you might say, well, that must be because really Mexico is admixed and people have more European ancestry, let's say in the North, and more Indigenous American ancestry in the South. And so you're just visualizing the differences between those two. So maybe if we just capture the frequencies in Indigenous American ancestry versus in European ancestry, we would then be able to sort of expect this. And if we knew that someone was, mostly European ancestry, mostly Indigenous American ancestry, we would then be able to expect these different frequencies. And that's true for this variant I'm showing right here. That is why you see these frequency differences. The North of Mexico does have more European ancestry, and the South does have more Indigenous American ancestry. And that is what's driving the frequency differences here. However, I mean, you can see here, this is this plot, maybe not clear, but these are different farm GKB variants. And we're plotting their frequency in the European ancestry in the Mexico Biobank versus their frequency in the Native American segments in the Mexico Biobank. And so you can see that this variant right here is a very high frequency in the Native American segments and not very high in the European. And so that's why we get this geographic difference, North and South of Mexico. But there's more, because it turns out that if we look only at the indigenous ancestry in Mexico, and we can do that using these techniques that we saw at the beginning, where we can label every segment. To go back too far, I'm going to regret this, but looking at every segment, that's only the segments that are European ancestry or only the segments that are Native American ancestry using this local ancestry analysis. So we can do that across the entire Mexico Biobank, and we did. And looking only at the segments that are Native American, we can calculate the frequency just in Native American segments across every region of Mexico for these different farm GKB variants. And what we see, now I've gone too far again. What we see is quite a bit of variation, even in the Indigenous American ancestry. So this shows at the state level for this particular variant, a lot of variation just in the Indigenous American ancestry. And so it's not enough actually to ask, is somebody mostly European or mostly Indigenous American if you're trying to look at these frequency differences. For some of these variants, it matters whether they are coming from Mayan Indigenous American ancestry, which is what you have here in the Yucatan, and that's where you have these really high frequencies, or whether they have more Mesoamerican, like Aztec type Native American ancestry from the center of Mexico or the north. And so it actually matters even in a very regional sense. And this is because of founder effects, which I alluded to earlier. So you have these founder effects, different groups, which really randomize allele frequencies and can really make a difference even on a very local level. And we see this in Polynesia as well. When you have each island settled by a small number of founders, you have very large frequency differences in Hawaii versus in the Maori in New Zealand, or in Samoa versus in Easter Island. And we see the same thing in the Americas. Strong founder effects, very different frequencies for those with Mayan Indigenous American ancestry versus those with more Northern Mexican or Mexico City, like Aztec or Navajo indigenous American ancestries. And this will, of course, play out in terms of even individuals who are now living or who have been long living in the United States, Hispanic individuals, wherever they come from, whether it's from the Caribbean or from Puerto Rico, Dominican, or from Central or South America. The indigenous American ancestry is going to be quite different from each of those regions, and it's going to have different allele frequencies. So we're really not going to capture that with a category that just calls everyone Hispanic who comes from these sort of regions. And so it's a role that I think genetic ancestry has to play in some of this. And so this is, a map. There's going to be a database that we're putting together that gives you this sort of for every variant. You can just visualize it across the different states in Mexico. I don't have time to talk about founder populations in more detail, but as I mentioned, it's really key and it's not just islands. It's throughout the Americas. It's throughout colonial small numbers of founders that had really strong effects in randomizing allele frequencies. You can think of the Pennsylvania Dutch and the Amish. And then there are founder populations that are very important in Europe as well. You can think of the Ashkenazi Jewish population, which has now been shown to have come from an effective population size of founders of around only 300 in the Middle Ages. And these have really strong genetic effects. And we understand some of these groups at some level, like the Ashkenazi Jewish. We know some variants that are important there and that are characteristic. And we don't know very little about others, including Pacific Islanders, including indigenous American populations. I'm showing this again, allele frequencies in Mayan groups versus in northern Mexican groups, et cetera. And so there's still a lot of work to do there. But these bottlenecks can have really large, and here you see every island kind of different frequency effects. And this is an allele that we found was actually really important in the pandemic, and is at quite high frequencies in a few of these islands due to these founder effects. And there's all sorts of other such things going on, and that are probably of interest to ClinGen. And I won't go through this in too much more depth, but we're still looking into all of these, as I mentioned, populations across many founder populations across East Asia, Oceania, and the Pacific, trying to characterize these founder effects and these allele frequency differences. So I want to thank all the participants. You recognize this plot here. This is these local ancestry plots. So we return them. It's really important to return information to the participants who are actually the ones providing all this data. And so we went back to these regions to return some results to all the participants, and really appreciate all of them, and appreciate all the students who worked with me on these different algorithms. And it's really been great working with students from many different places, something that Zoom has allowed us to do, and it's been a lot of fun. So yeah, any questions? I know I'm getting close to the end here, but hope you enjoyed.
Thank you, Alex, for an incredible talk. I know we're running into time, so anybody who has to leave, please feel free to. If you do have questions you'd like to ask them now, please feel free to raise your hand or add them in chat. Otherwise, if you email them to me, I'll put my email in chat as well. I can make sure we get them to Alex.
Alex, I was just going to say thank you as well. And that we obviously have a number of discussions that we need to move forward now that positions have been settled. So after you're through teaching this quarter, I think it'll be important for us to move some of those things forward. I was just going to recommend that, given the time we are over, is if people do have questions for Alex and would like to see them discussed, please email them to Clarissa, and she'll get them to Alex and make sure that the answers get disseminated amongst all of us. So Alex, again, I want to thank you on behalf of everybody. You gave a great talk. We could have given you many more hours. And so maybe that's something we can think about in the fall, is having, sort of the second half of all the things that you couldn't cover today that, you might be able to cover then or in the winter. So thank you, everyone.
Thank you, Terry. And I just do want to credit Terry. I mean, thank you for putting this up. This is an idea, this sort of frequency things that originated from a discussion I had with Terry. I'm really looking forward to working with you more on this as we advance it, Terry. I think it's really exciting to look at these sort of frequency differences. So.
Yep. No, I think it's great. Okay. Thanks, everyone.
Thanks, everyone.
Anything else or no?
Nope. Have a great week, everybody.
Thank you, everyone.
Thanks.